So it's my distinct pleasure to introduce Dr. Ron Kostanji. There's an effort going on now at the University of New England to kind of focus on all things aging, which is like a dream for me. But many of you know that we have received $1 million donation from a board member to start a Center for Excellence in Aging and Health. And we are just about completing the job description for that. It will be someone who has probably R01 NIH, NIA funding from the National Institute on Aging. This will be a research center, and so we want to keep moving in that direction. And then not long after that, weeks after that, we received the international designation as an age-friendly university. We were the 5th university in the U.S. to receive this designation. So that was pretty cool. So yeah, it's way cool. Dr. Dora Mills, who is our interim vice president for research at UNE and also the director for the Center for Health Innovations, Center of Excellence for Health Innovations. Through as her role as VP for research, she is taking a pretty large chunk of money and creating an opportunity for faculty mini grants all focused on aging. And that announcement will probably be made in October to open up the request for proposals. So there's a whole effort going on around aging, which is just like, I love it. It's great. So with that, actually, Ron and I have talked on the phone before, and now this is the first time we're meeting. And just talking with him on the phone, I was just like, I love this guy. He's just so cool. So when we were looking at staying along the aging theme, he was the first one that came to mind. And he graciously accepted our invitation. So to read his bio, so Ron Kostanji received his PhD in genetics at Utrecht University in the Netherlands, working on the genetics of atherosclerosis. He continued this work as a postdoctoral fellow with Beverly Pagan at the Jackson Laboratory in Bar Harbor. He moved back to the Netherlands in 2004 to study the genetics of kidney disease at the University Medical Center at Groningen, but after a few years decided to return to Bar Harbor. Who wouldn't, right? I know. Dr. Kostanji is now an assistant professor at the Jackson Laboratory, where his main focus is on the aging kidney. In addition to his position at the Jackson Laboratory, he is an adjunct faculty at Tufts University School of Medicine and the University of Maine, where he currently teaches a graduate course in biology of aging. Dr. Kostanji has published 76 peer-reviewed journal articles, several book chapters, including a chapter in the new edition of the Handbook of Biology of Aging. It's also important to note that Jackson Laboratory is one of 5 Nathan Schock Centers of Excellence in Biology of Aging recognized by the National Institutes of Health. So, and we are the only ones up in this whole northern New England area, Texas and California. I forget where the others are. So please give a warm welcome to Dr. Kostanji.
Am I? Yeah. All right, thank you. So an hour ago, there were a lot more people here. So I'm just wondering if it's me or if it was because of the free beer. So thank you very much for inviting me. This is actually the second time that I'm at this beautiful campus. The first time was 2 years ago when I was running the Maine Coast Marathon. And it was a really hot day that day, and I was just struggling to get to the finish line. So I hope today is going to be less of a struggle. This afternoon the talks were just like really great, and the poster presentations also. So like the bar is really high. So I hope I can interest you in the work that I've been doing in the last few years at the Jackson Lab. And so I think in the last few years, there's really been an increase in aging research in Maine. So institutes like MMCRI, University of Maine, Mount Desert Island Biological Lab, the Jackson Lab, and of course also here the University of New England are increasing their aging research. And I think that we can make it even stronger if we would collaborate. And I think the first step in that collaboration is first to learn from each other what we're doing so that we can see if there's any overlap where we can really work together. And so that's why I'm presenting here today some of the work that my lab is doing, but also our Nathan Shark Center of Excellence in the Basic Biology of Aging at the Jackson Lab, to show you what we're doing and maybe to see if there's some overlap there. So as already been mentioned, so we're one of the actually 6 Nathan Shark Centers in the US. It changes every 5 years. Like, so there's this funding cycle, 5-year funding cycle. 15 years ago, the National Institute on Aging decided that they wanted to create these centers, and so their mission is, and I'll just read it, to enhance the quality of research in the basic biology of aging, to facilitate the planning and coordination of research on aging activities, establish potential regional or national technological and intellectual resources in gerontology, and to provide a suitable environment for junior faculty and other investigators new to aging research. And so the Jackson Lab, together with the Aging Center in Seattle and in San Antonio, are the 3 centers that have been a center since the inception 15 years ago, and so some other centers been coming and going. Each center has its own focus, and that really depends a little bit on the investigators that are involved in these centers. And because of the Jackson Lab has an almost 90-year focus and expertise in mouse models and genetics of mice, it's obvious that we're working on the genetics of aging using mouse models. So, as probably everybody knows, aging is a primary risk factor for many common diseases. And so what we're interested in is to try to find the genes that are involved in this aging and make different organs more susceptible to disease. And an approach that has been used over the last 15, 20 years is doing these genetic studies in humans, where we're trying to find genes that are associated with longevity or age-related phenotypes. And although they're kind of successful, really it's a little disappointing on what they find. What we see is that every time with these studies, really only 2 genes keep popping up. That's ApoE and specifically ApoE4, which is associated with cardiovascular disease, and we also know that ApoE4 is a risk factor for Alzheimer's disease, and FOXO3. And so, because these kind of like genomic studies are kind of like disappointing, a number of years ago we decided to not look at the genome, but to look at the transcriptome. And so we were just a small part of this big CHARGE consortium. It's a consortium of human cohorts in both the US and in Europe, where a lot of effort been put into try to get all the protocols in the same way and to measure the same things, so that we were able to combine these different cohorts and so get a large enough population size that we can do actual real nice studies. And so in this big cohort, we looked at 15,000 whole blood samples from people between 17 years old and over 100 years old, and then used microarrays to look at gene expression in these whole blood samples. And so we identified about 1,500 genes that were differentially expressed with age, and there were about 800 genes that were down-regulated with age, and about 600 genes that increase in expression with age. And so this is really interesting, but the problem is that, of course, this is going to be a mix of genes that are really driving these aging processes and are really involved with aging, and genes where the expression is just a result of this phenomenon. And so really figuring out what cause and consequence is very difficult to do in human studies. And so the approach that we took in my lab was to take these candidate genes that were found in humans and then test them in a lower organism that has the benefit that it has a very short lifespan, is very cheap, and it's really easy to manipulate these genes in these lower organisms. And so to screen these genes for effective lifespan in these lower organisms. And in this case, we picked the worm C. elegans. Now, one hurdle in that is that we need to figure out what are the orthologous genes in the worm for these candidate genes that we have. I mean, obviously, not all human genes are present in the worm genome, and it's sometimes very difficult to figure out what the orthologous gene is in the worm for these human genes. Over the years, a number of tools have been developed to try to predict what the orthologous gene is in another species, and some of them, and each tool has good things and bad things, but still the individual tools are not really good at predicting the orthologous genes. And so we developed this new tool that uses all these different tools and then use machine learning to come up with a meta predictor. And we've shown that this tool has a really high confidence in predicting the orthologous genes. And so this tool now is online and can be used not only to predict orthology between human and worm, but also between many different other species. And so what we did was we took the 125 most significant genes from our human study. We looked at the orthologous genes, and we were able to find 88 orthologous genes in the worm that corresponded to 61 human genes in our data set. And then there were a few genes for which we could not knock down the gene in the worm. That's the next step that we wanted to do. So in total, we actually tested 82 genes from our original data set. And so doing lifespan studies in C. elegans in the worm is really easy. We take about 100 worms that are in the L4 stage, so in the adult stage, and we grow them on plates. We keep them on plates that contain this chemical fodder, and that's to prevent them to lay eggs, because otherwise you would end up with a plate with worms with all different ages, and you don't want to do that. And then knocking down a gene in C. elegans is really easy. C. elegans' favorite food is E. coli, so we keep these worms on plates on which we grow E. coli. And if we want to knock down a gene, we give these E. coli a plasmid that expresses double-stranded RNA for our target gene. And so our worms eat the E. coli, they ingest the double-stranded RNA, and then in their cells it's being processed to these siRNAs that then bind to the target messenger RNA. Messenger RNA get degraded, and so the protein is not being formed. And so, as I said, we take about 100 of these worms, and then every day we just see how many are still alive. And so the lifespan curve that you see, so starting out with 100 worms, the black curve is kind of like the normal wild-type worm. And so you see this lifespan curve, and around almost 40 days, all the worms are dead. And then when we knock out a particular gene, if they indeed have a lifespan effect, you see a shift in the curve. And so testing our 82 genes, we tested them both at 15 degrees and 25 degrees, and it's really important to test both temperatures because it has a significant effect, as you can see in the results. When we did this at 15 degrees, we only had 5 genes that had a significant lifespan effect. And when we tested at 25 degrees, we had 36 genes with a significant lifespan effect. The one gene that stuck out that has a more than 20% increase in lifespan, both at 15 degrees and 25 degrees, is this Kinu1. And Kinu1 is an enzyme, it's called kynureninase, which is in the kynurenin pathway. And so we show that both when we knock down the gene, and we later confirmed that by actually also knocking out the gene, so making a mutation in the worm genome, it has exactly the same effect. We get a more than 20% increase in lifespan. Now, the kynurenin pathway is really interesting in terms of aging because it's associated with a lot of age-associated diseases. So it's been shown that it's involved in neurodegeneration and cognitive impairment. It's associated with chronic inflammation, cancer, cardiovascular disease, diabetes, and also kidney disease. In fact, so my lab's major focus is kidney disease. And we already identified the kynurenin pathway a couple of years before we actually found this effect in worms. So a few years ago, we did a genetic study on outbred mice, testing them for albuminuria, which is a marker for kidney disease. And we found that there was an association between albuminuria, so kidney disease, and the gene KMO, which encodes an enzyme that's just one up from kynureninase in the pathway. We showed that in healthy humans, in a healthy human kidney, KMO is expressed in the glomerulus, as you can see in the upper panel. And people with diabetic nephropathy have a decrease in KMO expression. To see if this is causal, we knock down KMO in zebrafish, and to measure whether they have kidney disease or not, we have to do a trick, because in humans and in mice, of course, it's pretty easy. We collect some urine. In the case of the mice, we just pick up the mouse and hold it above a cup. And the first thing a mouse does when you do that is they pee. And so then it's really easy to just measure albumin or protein, which is a marker for kidney damage. Because in the glomerulus, in the filter of the kidney, it's too small to let proteins or albumin through, and so normally it shouldn't end up in the urine. But when you have kidney damage, that filter is broken, and so the albumin is able to pass through the filter and end up in the urine. Now, the trick that we use with zebrafish, because of course we just can't collect urine from these zebrafish, is that we have a transgenic zebrafish that has this fluorescent protein that's in the circulation. And because there's little veins in the eye, we can measure the fluorescence in the eye. And so in the top panel, you can see a wild-type zebrafish, so it has a nice fluorescence in the eye. Now, the middle one is where we knock down KMO. And so if there's kidney damage, this fluorescent protein is leaking away in the water, and the fluorescence goes down in the eye. Also, below, I know it's hard to see, are some electron microscopy pictures of the zebrafish kidney. And what we see is that in the left, we still see a nice glomerular basement membrane and podocyte food processes, which is the filter of the kidney. And on the right side, we see that these podocyte food processes have been detached from the glomerular basement membrane. And so that's what causes the albumin to leak in the urine. We also confirmed this in the mouse, so we made a knockout mouse for this gene, and also the knockout mice show already from an early age, both in females and males, high albuminuria in the urine. So going back to the worms, we were interested in the other enzymes in the pathway, so we systematically knocked down every single enzyme in the pathway and showed that knocking down every single enzyme leads to an increase in lifespan. Now, the next question was, is this a common mechanism? So is this lifespan effect caused, doesn't matter which enzyme we knock out, it's all the same mechanism, or are these different mechanisms? And this is the full pathway, and what you see is that there's a lot of different side branches in the pathway. On the lower end, we see NAD+, which is connected to the mitochondria, sirtuins, hypoxic response. Kind of like in the middle, it's connected to insulin and the IGF-1 signaling pathway, which has been associated with lifespan and aging. And in the top part, we see that it's connected to the mTOR pathway in dietary restriction, which we also know is associated with aging and lifespan. So to kind of like get a handle on that, what we did was we combined our knockdowns of the enzymes with mutations in worms that we knew had an effect on different pathways that are associated with aging and lifespan. So on the left side, we had worms that had a mutation in DEF16. They live longer because they have a defect in the insulin signaling pathway. The middle one is a mutation called E2, and that mutation causes difficulties for the worm to eat, so it's kind of like a dietary restriction model. And then the lower one is RSKS1, which is a mutation in the mTOR pathway, which we also know is associated with aging. And so by combining our mutations with these three different mutations, what we expect is that if our enzyme plays a role in the same pathway as, for instance, DAF16, like you see in the upper left, then there's no difference in the lifespan curve. But if they're in different pathways, for instance, HAO1 and DAF16, then you see that in addition to the lifespan effect of the DAF16 mutation, you see an additional effect because of the knockdown of HAO1. And so you can see that the three different enzymes that we tested all show a different pattern. And so this suggests to us that it's not a common pathway, but that different mutations in the pathway lead to different mechanisms of lifespan expansion. The other thing that we were interested in is to see what the effect was on the different metabolites in the pathway. And so using mass spec, we just measured all the different metabolites in the pathway. You can see in the top panel when we knock down TDO2, which is the enzyme all the way up in the pathway, we see an increase in tryptophan, which makes sense because you cut away the enzyme, so you just get a buildup of the substrate. The same effect you see when you knock down Kino1. You see that 3-HK and kinorenin are increased. And when we knock down HAO1, we see an increase of 3-HAA. Now, seeing these metabolites go up made us wonder what would happen if we would just add the metabolite into the media of the worms. Would this have an effect on lifespan? And the answer is yes. When we add different concentrations of 3-HAA, we see an increase in lifespan that increases with the dose of 3-HAA we use. And in fact, when we use 1 millimolar of 3-HAA, just adding it to the plate, we see that the lifespan effect in the worms is the same as when we just knock down the enzyme. And this also confirms the different mechanisms of aging. So when we knock down IDO or TDO2 or the kinoreninase, we see a lifespan effect, but then if we also add 3-HAA, we see an increase in that lifespan effect. So what we're currently working on is trying to figure out how 3-HAA affects aging in worms. So one thing what we're doing is to see where does the 3-HAA end up in the worm. And fortunately for us, the nice thing is that 3-HAA has a red color while all the other metabolites are colorless. And worms are pretty transparent, so it's pretty easy to see where the 3-HAA is going. Also, so far we've done kind of like whole body knockouts, so where the enzyme that we knocked out was just knocked out in the whole worm, and now we're working on tissue-specific knockdowns to just see whether knocking it out in just, for instance, the muscle or another place, whether that already has the lifespan effect, or if you really need to knock it out in the whole worm. There's been some publications of 3-HA being involved in oxidation and being an antioxidant. And so we're testing that at the moment. So we're looking at the things that are being oxidized and also what the effect is on the worms if we put them on plates where we increase their oxidative stress. For instance, adding paraquat to the plates. And finally, we're looking at protein homeostasis. So there's a couple of papers where 3-HAA could be beneficial. And for instance, it can bind to beta amyloid and prevent the aggregation of the protein in vitro. And to test that, we actually have a couple of really nice worm models. So this is one of the models that we have. This is a Huntington's model. And so it has a fluorescent polyglutamate, which in young worms, and it's expressed in the muscle. And what you see is that as the worms get older, you see this protein aggregation. So you see these aggregates forming in the muscle cells. We can measure those aggregates, and we haven't tested it yet with the 3-HAA, but for instance, if we knock down TDO2, we see that there are still aggregates, but the number of aggregates and the size of the aggregates is a lot smaller. And so this is a model, and we also have an Alzheimer's model that we're going to test. So, of course, doing this in worms is great, but really doesn't have a lot of application in humans, which is, of course, going to be our final goal. And so we're now moving it back from the worm. Now that we tested our genes, we know which ones have an effect on lifespan. Now we're moving it back to a mammalian model to study these genes in mammals. And so what we've done is for the 6 most significant and also a number of genes in the Skinner-Renin pathway, use a CRISPR-Cas9 made knockouts for 10 genes. We have the animals on the shelf at the moment, and they're ready now to go into this aging pipeline where we both study lifespan. So this is going to be a long study because mice live about 3 years, and if we indeed have lifespan extension as we have with the worms, then it could easily go to 4.5 years. And in the meantime, we also at different time points, initially 10 months and 20 months of age, we have these animals go through a battery of phenotypes. For instance, we look at behavior, grip strength, kidney function, hematology parameters. We look at glucose, insulin, cytokines, liver function. We look at cataracts and urinary incontinence. As for 3-HAA, what we're going to do is we're going to compare wild-type mice with mice that we are going to treat with 3-HAA, mice that we're going to treat with 4-CL-3-HAA, which is an inhibitor of the enzyme, and we're going to compare that with the kynureninase knockout and the HAU knockout. And then those go through the same pipeline, but also we're going to look at more specifically at aging effects in the brain, the heart, the kidney, immune system, and kind of also, depending on what we find in our worm studies, take a closer look at oxidative stress, proteostasis, and some specific age-associated diseases. Kind of like shifting gear a little bit, as I already mentioned, really like the focus of my lab is kidney disease, and specifically the aging kidney. And why I'm interested in the aging kidney is because I think the kidney is a really good model organ for aging. We know that age-associated changes in the kidney are easy to detect, both in humans and mice. We can just draw some blood, collect some urine, and that already gives us some readouts for function and damage. We also know that relative to other organs, the aging process starts early in the kidney. And also the kidney has an enormous effect on aging and age-related disease, other age-related diseases in other organs. For instance, people that are on dialysis, in the first year that they're on dialysis, their risk for a cardiovascular event increases by 50%. Their risk for Alzheimer's disease increases by 40%. And so it's very likely that there's a big link between the aging kidney and aging of other organs. We see that as we age, our kidney function is declining. This happens earlier in males than in females. We know that things like hypertension, diabetes, smoking increases the steepness of the curve. And at some point you reach this level where if you go below this level, you have kidney failure and you need either a new kidney or you need to go on dialysis. At the moment in the US, there's over 500,000 people on dialysis. It's, of course, an incredible burden if you have to go to a dialysis center 3-4 times a week and being hooked up to a machine for about 4 hours at a time, and especially, for instance, in Maine, where dialysis centers are not not next door. That's an incredible burden. It's also incredibly expensive. So a dialysis patient treatment costs about $100,000 a year, and that's excluding medication. And so we're really interested in trying to identify the genes and the pathways that are underlaying this curve here, because if we are able to kind of like manipulate those pathways and decrease the steepness of this curve, then that would already be an enormous gain. Just one of the things that we have been really interested in the last few years is something called mesangial matrix expansion. And so in our kidney, we have about 1 million filtration units called nephrons. And each nephron consists of a glomerulus, which is the filtration unit. That's where the blood is being filtered, and you get the filtrate. We produce about 180 liters of filtrate every day, and so fortunately we have tubuli that resorb 99% of this filtrate, otherwise we would be in the bathroom the whole day. If we take a closer look at the glomerulus, that's kind of like on the lower left, it consists of these capillaries where the blood flows through, and then the outer ring is the glomerular basement membrane, and then podocytes, which filter the blood and so form the filtrate. In between these capillaries, so in the middle, that dark in the middle, are mesangial cells. And these mesangial cells, they produce mesangial matrix, which is responsible for maintaining the structure of the glomerulus and also regulating the blood flow. Now, in a normal healthy kidney, there's this balance between making this mesangial matrix and breaking it down. So you get this constant turnover. But as we get older and people that with diabetic nephropathy or lupus nephritis, there's this disbalance. And so they start to produce more matrix than is being broken down. And so you get this mesangial matrix expansion. And so on the right side, on the upper right, we have, this is a healthy, normal mouse glomerulus. And on the bottom, you can see a glomerulus with mesangial matrix expansion. So you can see that there's a lot more pink in this glomerulus. And that causes a decrease in function, in filtration function, and it also causes inflammation. And it's kind of like the beginning of the end. So we did a big study in looking at mesangial matrix expansion in aged inbred strains. And so first, just want to put in one slide to just make sure that we're all on the same page with our mouse genetics. So an inbred strain is when we brother-sister-made mice for more than 20 generations. And so the result is that within an inbred strain, every mouse is genetically identical. There's no genetic variation between animals from the same inbred strain. So making inbred strains started in about 1909 when C.C. Little started to make inbred strains. First inbred strain was a DBA2, which at the Jackson Lab we still have. And then he moved on, creating more inbred strains like the B6, which at the moment is the most used inbred strain in the world. And then in 1929, he moved on and founding the Jackson Lab. And so nowadays we have thousands of different inbred strains. And as I said, there's no genetic variation between animals from the same inbred strain. but there's a lot of genetic variation between different inbred strains. And as you can see here, right, these are all different inbred strains. They're all in the same environment. They're in the same mouse cages. We give them the same diet. But you see a lot of variation in size, in coat color. You might even spot some differences in behavior. And if we look at mesangial matrix expansion, and these are 20-month-old males, we also see a lot of variation in this mesangial matrix expansion. You see that on the left, there's inbred strains that have a lot of mesangial matrix expansion, and on the right, strains that almost have no mesangial matrix expansion at 20 months of age. And so basically, the question that we asked was, what is genetically different from between the mice on the left and the mice on the right? So for most of these inbred strains, we now have the complete genome sequence, and so we can just like compare the genomes and see what's different. What do the strains on the left have in common and is different from the strains on the right? And so when you do that genetic analysis, we find one single gene that determines this difference. This gene is called FAR2, and it encodes an enzyme called fatty acyl-CoA reductase 2. And what we found was that all the strains that develop mesangial matrix expansion have this 9 base pair sequence in the beginning of the gene in what's called the 5' untranslated region, so it's just before the coding sequence, that is absent in all the strains that do not develop mesangial matrix expansion. Also, we noticed that all the strains that develop mesangial matrix expansion have a 2-fold higher FAR2 expression than the strains without mesangial matrix expansion. And so we did a number of studies. We showed that this presence or absence of the 9 base pairs is directly responsible for the difference in expression. We identified that it's a transcription factor binding site. We identified the transcription factor. We did a lot of different things. The one thing that we also did was we knocked out FAR2 in our mice. And what we see is that it doesn't prevent mesangial matrix expansion. When we look at 18-month-old mice, we still see that both wild types and knockouts, they develop mesangial matrix expansion. But what we do see is that this is postponed by at least 6 months. So if we look at 12 months, the wild types already have mesangial matrix expansion, the knockouts do not. So 6 months in a B6 mouse is about equivalent to 15 years in humans. And so being able to postpone this in a human for 15 years could have an enormous impact in progression to end-stage renal disease and having to go on dialysis. And in fact, we teamed up with a group at the University of Michigan who have gene expression of kidney patients and controls. And we see that indeed in people with chronic kidney disease, they have a higher FAR2 expression than controls. And specifically, if you look on the bottom, people with diabetic nephropathy and lupus nephritis have a higher FAR2 expression. These are 2 forms of kidney disease where mesangial matrix expansion plays a very important role. So I told you there's genetic variation between inbred strains, but actually, if you really look closely, there's not a lot of genetic variation. And this is because most inbred strains are actually derived from a very small group of mice originally in 1909, 1918. And so, although we are able to find some genes that are associated with age-related kidney disease, we are worried that we're missing out on some genes just because there's no genetic variation between our mouse strains. And so to solve that problem, we developed a new genetic mouse resource, which is called the diversity outbred mice. What we did was we picked 8 inbred strains that we knew is incredibly diverse, genetically diverse, by picking strains that were derived from different mouse substrains. So as you can see, most inbred strains are derived from Mus musculus domesticus in that blue circle. And so what we did was we also picked some inbred strains that were derived from Mus musculus castaneus and Mus musculus musculus. And so when you combine those 8 strains, there's about 45 million segregating genetic variants, and that's more genetic variation than that's in the human population if you not include rare variants. And so we started mixing up these 8 founder strains, and so creating an outbred population where each mouse is a unique combination of these 8 founder strains. And so using that cohort, we did a very big aging study. So we did a longitudinal study where we just kept 300 males and 300 females on the shelf. Every 6 months, we collected urine and blood. And other than that, just we left them on the shelf until they died of natural causes. And the interesting thing that we see is that if you look at the lifespan curve of an inbred strain, what happens is they're all alive, and then all of a sudden, in a short period of time, they all die, and all of the same disease. When we look at the lifespan curve in black of the diversity outbred, you see that it's more gradual, very much like a human population. And also, these mice live longer than most of the founder strains. These 2 mice here were called the golden girls. These are the 2 females that lasted the longest. They turned 54 and 57 months, which for a mouse is incredible old. We also did a cross-sectional study where we took 100 males and 100 females, and at 6 months, 12 months, and 18 months of age, so we had 100 males and 100 females for each time point. We collected tissues, and we collected urine and blood. And so this is now a resource that we're using at the moment. So what we've done is, so for 600 kidneys, we have the complete genotypes, we have DNA methylation data, we have gene expression data, we have proteomics, so protein levels for the 8,000 most abundant proteins in the kidney. We have renal histology, and we have urine measurements. And now we can really look at the relationship between these different layers of omics data. So we can look at what is the effect of the genotype on gene expression, what is the effect then on protein expression, and how does that translate to changes in the kidney in terms of histology, but also in urine. And of course, because we have these 3 time points, we can ask the question, what's different between 6 months and 18 months, for instance? And then once we identified some pathways that seem to be very important in the aging of the kidney, because we also collected different tissues and we're doing the same thing for those tissues, we can ask if this is kidney specific or whether this is something that's also happening in, for instance, the brain and the heart. And so this is something that we're working on at the moment. This shows you, for instance, the differences in gene expression between 6-month and 18-month-old kidneys. All the red dots are all genes that have an increased expression in 18 months compared to 6 months of age. And now we can identify which pathways are upregulated or downregulated. One very important pathway that's upregulated with aging is the actin cytoskeleton pathway. And our hypothesis here is that, so podocytes are very important, as I already mentioned. They're part of the filter in the kidney. And podocytes are not replaceable. When a podocyte dies, what needs to happen is that the podocyte next to it needs to make up for it. So it needs to grow, and it needs to kind of like move over. And so this requires upregulation of the actin cytoskeleton to be able to cover for the dead podocyte. And so with this knowledge now, our prediction is that if we would take an 18-month-old kidney that has the gene expression profile in the actin cytoskeleton pathway of a 6-month-old kidney, then we would predict that that kidney looks very healthy, right? That it's a healthy kidney when we look at the histology. While if we would look at a kidney that has the gene expression profile of an 18-month-old upregulated actin cytoskeleton pathway, we would predict that that would look as an old kidney. And so this is exactly what we did. We just selected a number of kidneys with a normal 6-month-old actin cytoskeleton gene expression profile and one with an 18-month-old profile. On the left, you see one that had a 6-month-old profile, And this indeed looks like a very healthy kidney. We see a nice glomerular basement membrane with nice podocyte food processes. So maybe I can point them out. So glomerular basement membrane, you see these nice little food processes. And if you look on the right side, this is a kidney that had an 18-month-old gene expression profile. And I hope you can see that like the glomerular basement membrane is not just like one straight line, it's bulging. And also a lot of the podocytes just being what's called effaced, they're being detached from the glomerular basement membrane. So this is really exciting that by just looking at the gene expression profile, we can actually predict kind of like the age of the kidney. The other pathway that we are looking at the moment that's really interesting is the senescence pathway. So cell senescence, of course, is a very important process in aging tissues. And we've seen in the last few years a couple of really nice experiments where they developed this mouse that, like, as soon as a cell goes into senescence, it kind of like self-destructs. So these mice cannot accumulate senescent cells, and these mice live a lot longer. Two important proteins in the senescence pathway are P16 and P21. And in our gene expression data of our diversity outbred cohort, we see that there's an increase in the expression of P21 and P16, and that's in the upper panel. If we look at our protein data, we see that there's a protein called ring1b that with age is going down. And this is interesting because ring1b actually regulates the expression of P21 and P16. The way it works is that normally at a young age, P21 and P16 messenger RNA are suppressed, so they're not being expressed because their DNA, so the DNA where these genes are located, is tightly wound around histones that are methylated. And so transcription factors just can't get to these genes to get expression. And this DNA this histone methylation is being maintained by what's called the polycomp repressive complex 1, and ring1b is an important component of that. So you can imagine that if ring1b expression goes down, so there's less of this protein, then it's no longer able to maintain the suppression, and as a consequence, you see an upregulation of P21 and P16. Cells start to senesce, and that's also the beginning of the end of the kidney. And so the thing that we're doing at the moment is, our idea is, what would happen if we could prevent this decrease in ring1b? What we would expect is that we would have fewer senescent cells in the kidney, and so we were able to maintain kidney function for a longer time. And so to do this, we just developed this new mouse. It's a transgenic mouse where we inserted a copy of RNF2, which is the gene encoding ring1b. We put behind it a fluorescent protein so that we can actually detect when our ring1b is expressed. So if we turn on ring1b, then we also turn on the fluorescent protein, and every cell that does that will glow green. We want to turn it on when we want to do it, so we want to make the decision whether there's expression or no expression. So the way we constructed it is between the promoter driving the expression and the actual gene, we put in a stop cassette. So in our mice, under normal conditions, this gene is not being expressed. It can only be expressed when we mate these mice with mice that have expressed the Cre enzyme. And the Cre is an enzyme that can recognize those lock sites, can cut out the stop cassette, and then the gene gets expressed. Now, this allows it also not only to decide when or where we express it, because we have mice where Cre is expressed using specific drivers, so we can only express the Cre in certain tissues. And in this case, we mated it with GGT1 Cre mice, so that Cre is only expressed in the proximal tubule of the kidney. And also, this Cre only works when we give the mice tamoxifen. So these mice express Cre, but Cre can only function when we give it tamoxifen. And so This picture here shows you a kidney of a GGT1 Cree RNF2 transgenic mouse, but we fed it corn oil, so the gene is not on, and there's no green. But what if we give these mice tamoxifen? The stop is being cut out, and now in the proximal tubal cells, this RNF2 is expressed. And so on the left, the dark is a glomerulus. There's no expression there. Including the fainter ones, there's some background, but are also tubuli, but not proximal tubuli. So that's where we are at the moment with these. So now we have a cohort of these mice. We have them on the shelf. We're going to wait till 12 months of age. Then we're going to turn it on, and then we'll see if that has an impact on our kidney function and the aging of the kidney. All right, so of course, in the end, we all want to go, want this to end up with the human, of course. So I've shown you now that we've been able to identify a number of enzymes, both using C. elegans, zebrafish, and mice, in different pathways that are associated with aging or age-related diseases, and specifically age-related kidney disease. Of course, a lot more research is needed, but we have the hope that with these pathways that we've identified, there's a couple of already existing drugs that are being used for other diseases, so that we can repurpose these to actually affect these pathways. And so hopefully that will help us to speed up the process of validating in the mouse to then applying this to humans. And this is actually, that's something that's already been happening in aging research in the last few years. So using worms and mice, a number of very important age-related pathways have been identified. We know about the insulin IGF-1 pathway, we know about the mTOR pathway, we know about sirtuins. All these have been identified in yeast and in worms, then have been tested in mice, and then several different compounds have been identified as being able to affect these pathways. And in fact, some of these are now being tested in humans, such as resveratrol, rapamycin. There's a really big study on the effect of metformin on aging in preparation, and then also acarbose. So with that, I want to acknowledge the people in my lab that are doing the mouse studies and the C. elegans studies, people of the Aging Center, and then on the right side, there's a number of groups that we've been collaborating with. The zebrafish studies, we're not doing that. We don't have zebrafish at the Jackson Lab. We do that in collaboration with Mount Desert Island Biological Lab. And on the bottom are like the mouse, the animal models that I like to play with. I talked to you about worms, zebrafish, and mice, and afterwards you can ask me about the fly and the black bear that we're studying. And with that, thank you for your attention, and I'll be happy to answer any questions.
All right, I'll start the ball off. Just C. elegans. No, that was just really an elegant talk, and I really appreciate it. And what I love is talks whenever I come up with all these questions, you answer it before I get a chance to ask you the questions. So I really appreciate the construct. Just for the sake, you know, so much of science is you're looking at the problem you're looking at because it's underneath the light post, and this is something that I won't go into a detail, but you're focused on the kidney. Now, until you said the conditional knockouts with just was it the proximal tubule?
Yep.
Okay. I would have said, what about the much more important phenotype that has to do with the heart? I do heart research, that's why. No, I'm kidding. But all sorts of systemic metabolism and heart and everything that is affiliated with chronic kidney disease. So my question is, it's not fair because you've done so much work with the genotype and the phenotype just of the renal structure, but do you have colleagues who are also looking at other comorbidities that are affiliated with end-stage renal disease, and maybe even in your mouse model.
So, sorry, so colleagues with...
Are you looking at heart? Are you looking at other metabolic
Right. So,
conditions?
right, and that's...
Yeah.
Sorry, yeah, so and that's really where, like, having an aging center really comes in nicely, where this is a number of scientists all having kind of like their favorite organ, but where we can combine the resources and use resources and then distribute them to the different investigators. So, for instance, in this diversity outbred study, we collected pretty much any tissue that you can think of. And so there's now different people studying these different organs. We still have the hearts, by the way, just letting you know. So, and the idea here is that, like, because we do all these studies on this same cohort of mice, on these same mice, that, like, in the end, we can actually combine all our data and then can look at, like, okay, so I found this in the kidney. What did you find in the liver or in the brain? All these mice went through behavioral studies. So we can make these links that you normally wouldn't be able to make just by just combining all this data and then just look at, like, relationships between things that you might not even think about. So yeah, so that is definitely ongoing. Not for all tissues. We still have freezers full of tissues. So if you have a phenotype or a tissue that you're interested in, please let me know. We'll be happy to share everything. And that's the other thing, right? So also all the models that we're creating, so this RNF2 transgenic model that I'm using now to look at the kidney, we'll be happy to share them. If somebody wants to cross this with a Cree to knock it out or turn it on in the heart or in the liver, then that's definitely possible. I mean, and that's really one of the missions of the Nathan Shock Center, to really make all these resources available to investigators within JAX, but also outside of JAX. Yeah.
Well, before you say, tell us something about the bear.
Ha! All right. So the bear. Do I have another half hour? The bear is interesting because when bears go into hibernation, they turn off their kidneys. And so during hibernation, their kidney function decreases and they develop damage. And so when a bear wakes up from hibernation, it basically is a dialysis patient. But unlike dialysis patients that cannot recover, bears just recover within a couple of weeks. So if you look at the kidneys of a bear, let's say 2 months after hibernation, they just look perfectly fine. And so we're really interested in how can they do this. And so we've been collaborating with the main tribes to collect kidneys. So in 2 years, we've collected about 70 kidneys before and after hibernation. And we did gene expression studies. And so we did a gene expression analysis to see which genes are differentially expressed before and after hibernation. And so we identified about 160 genes that are indeed differentially expressed and might be involved in the recovery of these kidneys. And so now we're making mouse models to actually test that. So the way we do this is, let's say gene X, we found gene X, we make either we make or it's already available a knockout mouse for that particular gene, and then we induce kidney damage that you could say might be similar to hibernation. What we do is ischemia reperfusion. So we just do a small surgery, we clamp the renal artery for half an hour, then we unclamp it again, mouse just goes on, and then after a couple of weeks we look at the kidneys and see if there's a difference in damage between the knockout and the wild type. And so that's what we're currently doing. So doing ischemia reperfusion in a number of knockout models and see if that has an effect. But yeah, it's a fun project. I haven't been bear hunting, but
All right, Dr. King's going to kill me, but one more follow-up on that one. So bears in hibernation, I can't remember if they undergo significant muscle atrophy or they're protected against, but we have a lot of issues with aging and people being bedridden and astronauts being up in weightlessness. And I'm wondering if there's a collaboration with people who are interested in skeletal muscle atrophy and
So not
hypertrophy.
at the moment. So this is really only kidney so far, but I mean, so the contacts are there, and the tribes are really interested in helping us with our research. So there's definitely possibilities there. I mean, at the moment we only, and it's really, so the way it works is, so if you want to hunt bears in the spring, you can only do this on tribal land. And so that's kind of like how we get our samples, because we developed this collection kit and gave them to the tribe biologists. So when a hunter wants to hunt the bear, they need to ask permission from the tribe, and then the tribe says like, sure, but here's a kit. As soon as you shoot the bear, you need to get a sample. And so that's how, in a very short time, relatively easy, we got a lot of samples. And so you could do something very similar with other tissues. Yeah.
I wonder if you had done any work. I know it's not aging, but how would that compare to kidneys that are given for live donor transplant? So a person with one kidney, how would be their kidney condition after taking one
Right.
of their kidneys?
So, so I don't have a lot, so I'm not an expert in that. But it's usually assumed that, like, so you have overcapacity. So just taking out one, if you're healthy and you're taking out one kidney, then you're fine. However, right, of course, as you get older and you get more susceptible to kidney disease, it might be that, and I'm not sure if studies have been done, it might be that this curve is steeper for you than if you still have two kidneys. And one thing that might be involved in that is actually the number of glomeruli that you have. So there's a big variation in these filtration units between people. It's anywhere between 0.8 and 1.2 million. You can imagine that if you have 1.2 million, you can lose a lot more before you start getting into trouble than if you have only 0.8. The thing is, like, it's of course difficult to measure those because you can't just like take out the kidney and look at it. So it would be really helpful if we would have knowledge of that and would be able to measure, to count those before you actually donate a kidney, and so say like, well, you kind of like have a low number, maybe you shouldn't. So, but that's something that still needs to be explored. Yeah.
What caught my attention was when you said that the podocytes, they have to replace any damaged podocytes. So, and I would imagine that the podocytes in one single kidney would be enlarged to make up for the one that was donated.
Yeah, no, indeed, yeah. Thank you.